Thank you very much, Will, for agreeing to give a Build a Cell seminar. And thank you for being the first speaker in our joint seminar series between the Build a Cell and the Biotechnology Institute. And you can get started. Thank you.
Great. Well, it's really exciting to be here and to get a chance to talk with you all and share some of our work. As Kate said, my name is Will Harcombe. I'm here at the University of Minnesota in both my tenure home as Ecology, Evolution, and Behavior, and then I'm also part of the Biotechnology Institute. And I guess as a brief introduction to myself, my first paper was actually in the evolution of snake coloration. So I started coming up through more eco-evolutionary biology, and then saw the light and how amazing microbes were and systems biology was, and so have steered more that direction recently. And let's see, let me just do this. Okay, so I'll jump right into it. So my group is really excited about microbial communities, and particularly the dynamics that come out of these microbial communities. So, not only are they beautiful, as you can see from this image of a microbial community pulled from dental plaque, but also they are critically important for everything from human health to global nutrient cycling. And as you all know, there's obviously a lot of excitement about harnessing the power of diverse microbes for a whole array of industrial processes, from energy production to bioremediation and degradation of pollutants. So because of this fundamental importance for biological and industrial processes, we really want to understand the composition and function of microbial communities, predict how those emergent properties will change through time, ultimately be able to manage microbial communities, and in the end, build some of these communities ourselves. So take a more synthetic approach, more similar to what you all are doing, but at the community scale. And as we start trying to tackle some of these challenges, there's some really fundamental questions which remain. Like, what is the scale over which individual cells interact? Who is interacting with whom in microbial communities? How do interactions between cells impact the response to perturbations? And how might these interactions shape not only the ecological response, but also the evolutionary dynamics in microbial communities? And so to address these challenges, we're taking what I call a bottom-up approach, but I'm realizing is distinctly different from the bottom-up approach that you all take. So for me, a bottom-up approach means starting with the genome sequence of an individual bacteria, and using that to understand something about the physiology of that organism. So what can it eat? What cool chemistry can it carry out? And then using that physiology within a cell to understand how different cells might interact with each other, whether they're going to compete or facilitate each other, for example. And then ultimately using these pairwise interactions to understand emergent community properties, so the composition and function of microbial communities. And then given my background, I'm also really excited about understanding how selection at each of these levels of biological organization feeds back to change the genomes that encode life. And as we try to sort of tackle this integrative challenge, my lab does a lot of sort of classic experimental evolutionary ecology in the lab. So setting up experiments with different species of bacteria that interact in different ways, and testing our ability to predict both ecological and evolutionary dynamics that will emerge from those interactions. And we pair these sort of wet lab experiments with many different types of modeling, and particularly this genome scale metabolic modeling that I'll touch on a bit at the end of the talk today. So I'm going to try and, I wasn't quite sure what would resonate with you all, but so I'm going to try and weave in a fair bit and give you sort of 3 brief stories about some of the questions we're trying to tackle, and some of the approaches we're taking. And hopefully that gives us a launching point for some discussion. So I'm going to talk about how location mediates competitive interactions, how metabolic mutualism influences the impact of antibiotics, and how metabolism can be used to generate emergent community properties. So launching into the first story, when we first started exploring microbial communities, what a lot of people do was to go out and sample a community of interest and then sequence it. And you come up with these amazing parts lists. And a lot of sort of the initial thinking was that sort of if you saw a species in your community, that meant it was interacting with everything else. But we know that's not true. We know there's some really intricate spatial structure in communities, such as the green bacteria that you can see only growing around the edges of this dental plaque. And so we really fundamentally, to understand these communities, need to understand what generates that spatial structure and which cells are actually interacting with which others. So I want to understand communities that look like this, but I'm not smart enough to tackle something this complex to start with. And so we wanted to take just the simplest possible scenario we could think of and threw some E. coli on an agar plate in the lab and wanted to see if we could, in the simplest of cases, predict interactions. So as any of you that have been in the lab know, you do this, you throw some bacteria on a plate, and you'll get some variation in the size of the colonies. So you'll get some isolated colonies, which are really big, and some colonies which are closer together, which are much smaller. So these colonies are clearly competing for diffusing resources, which are in the agar. And so we wanted to say, well, how well can we predict this variance in colony size as a function of the diffusion which underlies it? And this was a challenge which was tackled by an incredible grad student in the lab, Jeremy Chacon. And honestly, we thought this would take an afternoon, but it ended up being a little more complicated. So we went in the lab, we took images of a bunch of different plates of E. coli that had generated colonies. And like I said, we thought this was going to be really easy. We know they're competing for diffusing resources. So we thought if we just plotted the distance to the closest competitor against the colony size, that we'd be able to explain all of the variance in the colony size. And when we did that, we found that we were able to explain some of the variance, but there's still a lot of it that's left on the table. So we thought about it a little more and we thought, well, our problem was that we just took the distance to the closest competitor, and these things are diffusing, so we really should take the distance to all competitors on the plate, and that'll allow us to capture the rest of this variance. So we tried that, and our explanatory power went down. So we were left scratching our heads, but then ended up talking to some biophysicist collaborators, and they said, oh, we solved this problem a long time ago. What you need to do is consider not distances, but instead territory size. And once we did that, then we were really able to explain a gratifying amount of the variation that we saw in these colonies. And so what this means is that to predict the size of a colony, what you should do is divide the surface up into what are sometimes known as Voronoi areas or territory sizes, where the area for a given colony is the area on that surface, which is closer to that colony than to any other colony on the plate. So basically you can just draw a line halfway between any two colonies, and that creates a territory boundary. And one cool thing that comes out of this is that it means that geometry, not just distance, determines competition. So if we look at this focal colony here in red, its territory's boundaries are set by these open red circles. So this black circle, even though it's very close to this filled in red circle, actually doesn't impact the size of this red circle at all, while this open circle, which is a long ways away, does have a big impact because it sets a territory boundary. So once we understood this for E. coli going on rich media, we wanted to test how well that holds as we look at different species and on different nutrients. So we set up some very simple experiments, throwing throwing agar petri dishes onto office scanners in our incubator and track the growth of colonies through time for E. coli and Salmonella across different resources. And what we found was that there was a pretty big difference in how much variation we saw for, let's say, E. coli as we looked at it across different nutrient sources. And this is actually the coefficient of variation. So it's not just a function of how big the colonies get. It's really about the spread around that when you control for size. And so what we ultimately were able to understand is that on rich media, you end up seeing a lot of variance between different colony sizes. Whereas on poor media, like acetate, you see very little variance in colony size. And so on rich media, location matters a lot because you have these local interactions. So how close you are to another colony, sort of the geometry of your placement really determines how much growth you will have. Whereas if you're going on poor media, you have these global interactions. So all colonies are interacting with all other colonies and location becomes unimportant. What this fundamentally means is that sort of spatial structure is not a binary, but instead is this continuum, which is impacted by metabolism, by the nutrients which bacteria are eating. And digging into this, we're able to show that we could actually predict the importance of location by understanding the density and then the diffusion rate of metabolites and the growth rate. So a very simple equation helps us understand how important location will be and the scale over which interactions will occur on surfaces. With this quantitative understanding of the importance of space, we're able to dig in a little further and uncover some things such as using that to predict when other types of interactions are occurring. So for example, when we looked at Salmonella enterica grown on glucose, we saw that there's deviation between the predicted size of colonies and the observed size. And ultimately, we discovered that this was because the Salmonella colonies were not only competing for diffusing resources, but they also ended up being inhibited by waste. So this plate turns yellow because the Salmonella are acidifying the plate as they grow, and this acidification halts growth as opposed to just running out of nutrients. Additionally, we found that you can think about evolution on these surfaces. And we were able to show that evolution, as populations evolve to grow faster, that adaptation means that their interactions become more local. And so as the growth rate of the ancestral population increases, it takes longer and longer for a beneficial mutant to spread through that population. And this is fundamentally different from the expectation in liquid, where in liquid, what matters is only the relative fitness of mutants, not the absolute fitness. So we were excited about this observation that faster growth makes it harder and harder for mutants to invade in space while not in a well-mixed flask. So in summary of this first section, we showed that competitors are determined by geometry, that the scale of competition on surfaces is determined by growth rate, and that structure generates slowing rates of adaptation. So structure fundamentally alters the shape of evolutionary trajectories. So stepping into another brief vignette, I thought I'd tell you about some of our work associated with metabolic mutualisms, or cross-feeding, and how those might interact with antibiotics. As people study microbial communities more, one of the things that we as a field are realizing is that there is a ton of cross-feeding between different strains of bacteria. And by cross-feeding, I mean scenarios where one strain obtains metabolites from the excretions of another. Often these are things like amino acids. They can also be carbon, vitamins, all sorts of things are exchanged between different species of bacteria. And we thought that this sort of dependence on other bacteria might influence how bacteria respond to antibiotics. And a very simple verbal model, you can think about if you have an antibiotic which inhibits a blue bacteria but has no impact on a highly resistant yellow bacteria, if the blue bacteria and the yellow bacteria depend on each other, that antibiotic might indirectly inhibit the yellow bacteria by taking out the partner upon which it depends. And if this is true, it could be really important because we know that there's all sorts of cross-feeding between bacteria associated with the human body. Shall I answer questions as we go?
That's up to you. Some people leave it at the end. Some people answer as you go. Up to you.
Okay. That is a great question. So the question is, if you have a consortium with more than one species, would you still see the same pattern of colony size under low and rich medium conditions? And the answer is it's complicated. And that's something that we're currently tackling. But it's we've discovered that it's a lot harder than our sort of simple initial expectations. But I think there's some super exciting work to be done there. And yeah, I can touch back on that some more. So cross-feeding. We know that there's cross-feeding associated with the human body. So for example, a collaborator here, Ryan Hunter, has shown that there's cross-feeding in the lungs of patients with cystic fibrosis, and that the primary pathogen, Pseudomonas, cross-feeds off of anaerobes, which degrade the mucin. So there's this cross-feeding, which facilitates the invasion of a really important pathogen. And similarly, in the gut, we know that there are complex networks of species that interact to, for example, degrade polysaccharides. So a incredible grad student that I had the luck to work with, Dr. Beth Adamovich, set out to tackle this in the lab and used a cross-feeding community, which we engineered in the lab between E. coli, Salmonella enterica, and now Methylorubrum extorquens. And one nice thing about these species is that we've engineered in different knockouts so that they're exchanging known metabolites, where our E. coli relies on methionine secreted by our Salmonella and iron secreted by our Methylobacterium. Methylobacterium and Salmonella get carbon from the E. coli, and the Salmonella also relies on nitrogen. So we have known interactions between 3 different species. And because we know what they're exchanging, we can grow the bacteria in monocultures under the same nutrient conditions, where we're providing them with the same metabolites they're getting in community, but provide it so that they acquire them from the medium as opposed to acquiring them from other species. And we tested this with tetracycline among other antibiotics and did simple assays where we just dropped some antibiotic on a disc in the middle of the plate and measured the zone of inhibition, either when we were growing the species alone or when we were growing them in a 3-species obligate mutualism, obligate cross-feeding mutualism. So our question is, as we apply an antibiotic, if we know, how sensitive each of the 3 species is independently, can we predict the sensitivity of all species in this 3-species mutualism. So we first tested this for each of our species independently. Again, there's an antibiotic disc here. And what you're seeing is that the salmonella is highly resistant to the antibiotic, growing right up to the disc, while our E. coli and our methylorubrum extorquins are much more sensitive and are inhibited at much lower concentrations of the antibiotic. When we then looked in the 3-species case, what we found, it's a little hard to see here, but we found that the whole community was inhibited at a much lower concentration of the antibiotic. So even though the salmonella is highly resistant to this antibiotic, in the 3-species mutualism, it was inhibited at the same concentration as the methylobacterium. And so we use this to develop a very, again, super simple explanation, which we call the weakest link hypothesis, which is that if you're engaged in a network of obligately interacting species, the resistance profile of every species in that network is going to be set by the most sensitive member of that network, or the weakest link. So we were able to show that this worked in the lab, and we were then interested to test whether it had any applications outside of the lab. So we collaborated again with Dr. Hunter, who has developed this really nice system examining cross-feeding in the lungs of cystic fibrosis patients, and has shown that the primary pathogen cross-feeds off of anaerobes that degrade the mucin, releasing carbon, which the pathogen can use. So we set up a similar set of experiments where we first tested the resistance of methylobacterium to ampicillin in this case. The bars show increasing concentrations of antibiotic. And you can see that even at our highest concentration of antibiotic, we saw no significant reduction in the population size of the Pseudomonas, which is consistent with what's observed in patients where Pseudomonas is sort of classically difficult to knock back with antibiotics because it has such high resistance. In contrast, when we looked at just the anaerobes, they were dramatically inhibited by our lowest concentration of antibiotics. So you see, our lowest concentration significantly decreases the abundance of the anaerobes. We then set up a scenario where the Pseudomonas was cross-feeding off of the anaerobes, and we replicated our weakest link patterns, where now suddenly our lowest concentration of antibiotic is able to significantly decrease the abundance of the Pseudomonas because the antibiotic is taking out the weaker link of the anaerobes upon which the Pseudomonas is relying. We then were excited about asking, well, these interactions inhibit the immediate effect of antibiotics. Do they also impact the ability of bacteria to evolve resistance to antibiotics? So here we set up a simple stepping stone model where you can think about patches that have increasing concentrations of antibiotic. And we set up a model in which we allowed bacteria to grow in these patches, and they replicate with some mutation rate. So we pulled mutations from a distribution of potential resistance mutations. And as a result, over the course of growth, we get some genetic diversity arising within our population. We then engage in a dispersal step where we subsample from our population, which grew. So we just subsample into a patch with fresh media. And we also subsample into a patch with a higher concentration of antibiotic. We then kill off any cells that don't have enough resistance to survive in a patch in which they've landed, and then repeat the process where we again allow them to grow with a mutation. We then ran this simulation with a single species of bacteria or a co-culture of bacteria in which both bacteria are growing and acquiring mutations, but we set up a weakest link constraint such that a bacteria was not able to grow in a patch if its partner was not also able to grow in that patch. And what we see is that over the number of transfers, you can think about this as time or generations on the x-axis, that in all cases, we saw increases in the minimum inhibitory concentration of antibiotics of populations. So for example, if we look at just our one species simulations, the average drug resistance of bacteria in that population increased over time, as you would expect. So the bacteria are able to grow further and further out this gradient. As we then looked at simulations where we had either 2 or 3 species which relied upon each other, we found that the rate of antibiotic resistance evolution was much slower. So these multi-species communities in which the microbes were relying on each other, they evolved antibiotic resistance much more slowly. We then went in to test this mathematical model. So here we again used our cross-feeding E. coli and Salmonella. However, we just used the 2 of them. In this case, we dropped the 3rd species. And we tested the rate at which E. coli and Salmonella were able to evolve resistance to antibiotic when they were growing alone or when they were growing in a cross-feeding co-culture. We inoculated them on a 96-well plate, which had a gradient of antibiotics across the rows. And we set up multiple replicates of each of our treatments. We then transferred 10 to 20 times every 48 hours with shaking growth at 37. And our transfer regime followed that of our simulations. So what you're seeing here is there's growth in these first 2 wells. The numbers represent the concentration of antibiotics in that well. After 48 hours, we would subsample into a new plate where we sampled into a well with the same antibiotic concentration as well as 1 antibiotic concentration higher. And we did this for all wells. And then what we're testing is how quickly do populations evolve to be able to grow out this antibiotic gradient. And we tested this for both rifampicin and ampicillin. And what we found was that there was, consistent with our modeling expectations, a big impact on the rate of adaptation when bacteria were growing alone versus when they were growing in co-culture. So what I'm showing you here is, again, we have time on our x-axis, number of transfers, the average resistance, a measure of the average resistance on the y-axis. In this case, I'm showing you results for RIF. And you can see that our monoculture increased in resistance significantly, and that E. coli also increased in resistance when growing in a co-culture, but at a much slower rate than when the E. coli was growing in monoculture. We found that this held for Salmonella as well. So if we just focused on the Salmonella growing alone, it also increased its resistance much more quickly than the Salmonella did when it was reliant on E. coli. We then, in addition to looking at the rates of adaptation, we're also interested in the mechanisms of adaptation and whether relying on another species might alter the mechanisms through which populations evolve resistance to given antibiotics. And what we found was that it did have impacts on the mechanisms of adaptation. So I'm showing you sort of a cartoon of the targets of adaptation when E. coli was growing alone versus when E. coli was growing in co-culture, and the same for Salmonella. So we repeatedly, in monoculture, observed adaptations in genes associated with stress response for E. coli growing alone. However, in co-culture, we instead saw that adaptation was focused on genes which influenced the efflux of antibiotics out of the cell. So we saw this pattern, which is consistent with fundamentally different trajectories of adaptation as a result of a population growing in monoculture or when cross-feeding with another species. And we were particularly interested in these protein targets, which repeatedly acquired mutations in Salmonella when it was growing in co-culture, but never acquired mutations. We never observed mutations in that gene when Salmonella was growing alone. And this protein target is FTSI, so it's a penicillin binding protein that's involved in allowing the cells to divide. So it's part of the ring which allows cells to divide. And we repeatedly saw knockouts of this gene. And when looking through literature, we found that there's a great paper from a group in Spain, which has shown that this gene, FTSI, is really critical for allowing Salmonella to survive inside of macrophage. And in fact, what they showed is that if they knocked out FTSI, that it significantly increased the viability of Salmonella when attacked by the human immune system. So we were excited about this because it says that species interactions, so interactions between bacterial species, can drive bacterial populations to evolve down fundamentally different trajectories, and that those novel trajectories, which are enabled only in the presence of interactions with other species, that those trajectories can fundamentally change how that bacteria also interacts with the human immune system. And we then went back to our model to ask whether this evolution of different mechanisms is probably, whether we should expect that to just be something sort of specific to this system, or if it might be a more general phenomenon that we should expect broadly. And we think that it may actually be a broad phenomenon. So you can think about a population, and there could be some heterogeneity in that population, which I'm showing by these different colors of these cells. So the ancestral genotype may be white and have no resistance. There may be a number of cells that have a low level of resistance, and then very few cells that have these big benefit mutations, which cause them to be super resistant. And if this population is growing in monoculture and confronted with an antibiotic, we would expect that selection will favor the most resistant cells. So these really rare cells that have these big effect mutations, they are the ones that are going to drive evolution. And so you'll see that, that they're the ones that get selected. However, in co-culture, if there's no benefit of outpacing your partner, because if you're more resistant than your partner, you still can't grow, then we thought that evolution might be biased towards these more common mutations of smaller effect. And looking at our model, we found that that was true. So when we looked at the mutations which were sampled from the same distribution, when populations were growing in monoculture versus co-culture, we saw that in co-culture, our simulations sampled significantly smaller mutations of significantly smaller effect. So mutations which had a smaller effect on increasing resistance. So that was a lot. The idea was that antibiotics are more effective in mutualisms, that mutualisms slow the rate of adaptation, and that mutualism also alters resistance mechanisms. And finally, I want to take the last couple minutes and tell you about some of the simulation approaches which we're taking. So we're getting more and more genome sequence, but I think what many of us care about is biology, right? Like, what are cells actually doing? What are they eating? How are they surviving? And metabolism gives us a powerful way to go from genome sequence to phenotype, or genotype to phenotype. So as everyone's aware, we can create these genome scale metabolic networks, which incorporate all of the metabolic genes which are encoded in the genome of a cell. And then we can compile this list of metabolic genes into these networks, which show the cool chemistry that a cell can carry out. So the pathways, the biosynthetic and the the catabolic and the anabolic pathways that are inside of a cell. So stated again, if we have a genome scale metabolic network for a cell, we know all of the different metabolites a cell can take in, all the cool chemistry it can carry out, and what byproducts it might secrete as a result. So in other words, we can map out the global feasible physiology for a cell, all of the possible things that cell could do. And then if we apply an optimality criteria, which is known as flux balance analysis, we can take this global set of feasible physiologies and predict a likely physiology in a given context. So we can say, of our global set of feasible physiologies, which of these physiologies maximize, for example, growth? And because evolution acts on reproduction, we might expect that that is the physiology that a cell will have. And doing that, we can predict not only what cells can take up, but the proportion of resources that they will use and which specific resources they will use in a given environment. We can predict the pathways that will be active inside of a cell as it grows on those nutrients and what metabolites will be secreted. And there's been a sort of cottage industry which has grown up around using flux balance analysis. That can be really powerful for not only predicting the growth of bacteria in shaking flasks, but also bioengineers have been really excited about using this to think about how we could rewire some of these networks to secrete compounds compounds of interest. But we are interested in saying, can we use this to step beyond making predictions about single species to multiple species and potentially full communities? So we developed a platform called COMETS for computation of microbial ecosystems in time and space. And what we can do is we can start with genome sequences of multiple different bacterial strains. We can then optimize the growth of each of those genotypes independently. And we then use what's known as dynamic flux balance analysis. So we integrate through these optimization calculations. And as a result, we can predict what each cell will consume over a small unit of time, how much each of those cells will grow, what they will spit out. And then we take these excretions of cells and update the environment and do calculations again. And as a result, we can make predictions about how the excretions of one cell will affect another. We then also integrate this into a spatial platform where we allow diffusion of both metabolites and biomass. And as a result, we get these simulations of growth of multiple bacterial species on surfaces. And we get some really rich data out of this. We get not only quantitative predictions about how much of each species will grow, we also get rich data about exactly which pathways are active through both space and time, and the concentration of every metabolite in the system through both space and time. And so we can mechanistically understand how the interactions that we observe emerge from intracellular metabolism and nutrient gradients. We've shown that we can accurately predict the growth of single colonies on different resources, and that we can accurately predict spatial interactions. We've also shown that we can predict the interactions between species. So we set up models of the three species we use in the lab, and just introduced these known mutations into those models, and showed that we can not only predict that they would crossfeed and benefit each other, facilitate each other, but we could also accurately predict the exact equilibrium ratio of each of these species. And finally, because these are genome scale metabolic models, we can do fun things like going in and knocking out every gene one at a time in our community, and say, how will these different mutations impact the species ratios? And we've shown that we can make some accurate predictions about the impact of different mutations and about how interactions between species influence the genomic robustness of populations. We just released a new version of this called Comets 2.0. This was just a Herculean effort by Ilya Dukovsky in the Segre lab at BU, Georgi Bajic from the Sanchez lab of Yale, but now of Spain, and Jeremy Chacon from my lab. And some of the things that have been improved are improving the biophysical model for the spread of biomass. We've incorporated extracellular enzymes, evolutionary dynamics. We've enhanced the interface options. So there is a GUI, but there's also command line and Python toolboxes, as well as MATLAB. And there are extensive tutorials online. This program is all open source. It's freely available on runcomets.org, the email down here, the website list down here. And we hope that this will be a useful tool for people. And we're excited to collaborate with people to think about sort of additional things that could be done. So I hope that's given you some sense of the way that we are starting to try and take a bottom-up approach to understand the tangled bank of community dynamics, microbial community dynamics. Again, as brief conclusions, we first looked at the importance of location and showed that geometry and metabolism drive the importance of location. We showed that cross-feeding alters impact of antibiotic. Cross-feeding alters the rate and mechanisms of evolution of resistance to antibiotics, and that metabolism can be used to predict community dynamics. I want to thank some great collaborators, some amazing folks in the lab who are not only great scientists, but also up for wild things like curling and in a Minnesota winter, some funding organizations, and I would be happy to take any questions. And it looks like there are some great ones here.
Thank you so much. If you could read a question before answering it, that's going to go for the recording.
Sounds great. Let's see. So a follow-up question on colony size experiments. Could you engineer some selective transporter for a limiting nutrient that would rescue colony size diversity under low nutrient conditions that would generally result in small uniform colonies? Yes. So if you engineered a transporter in that allowed colonies to grow more quickly, then that would create that variant. So what's fundamentally important is basically the ratio between the diffusion of metabolites and the uptake rate of those metabolites. So if you engineered it such that the uptake rate increased relative to the diffusion, then yes, that would recreate the variance in colony size and make interactions more local. Yeah, great question. Let's see. Do you observe oscillation in the 3-organism system? Cool. Yeah, we do. We do initially. So they do converge to an equilibrium, but getting to that equilibrium takes multiple transfers. So we found that in the 3-species system, it takes about 3 transfers to get to an equilibrium frequency. But before then, there is oscillation in the species frequencies. Do you think that simpler genomes would exhibit similar evolutionary dynamics in co-culture? For example, a genome as simple as the 450 genes of M. laboratorium. Is this evolutionary dynamic you see a result of robustness of highly evolved E. coli, or would you expect to see it in simple engineered species too? Cool. Yeah, I would actually expect to see it in very simple species. I think that so we see it in our simulations, which have fundamentally very, very simple simple genomes. I mean, I guess we're not really thinking about the complexity of genomes in our simulations, and we see that this outcome occurs. So I think that for sure the rates I would expect to be different. The mechanisms, if you only have one, if it's so simple that you only have one mutation which provides resistance, then that would constrain the feasible mechanisms of evolution of resistance, in which case you wouldn't see different mechanisms in monoculture and co-culture. But to the extent that you have even a gene which can have multiple different mutations which provide some resistance, I think you would see many of these patterns repeated in very simple systems, and that it's not a impact of the E. coli genome necessarily. Follow-up, in the co-culture versus monoculture experiments, do you think complexity matters? Would you see similar effect if co-culture is between simpler bacteria and a eukaryotic cell, or would the dynamics change then? Yes, so the to the extent that both your populations can be inhibited by the stress, I would expect to see similar dynamics. So one way to think about it is that, in monoculture, as soon as a mutation arises, which provides resistance, it's going to sweep to fixation. And so you're going to just have this gradual climb in resistance. But in co-culture, if one cell starts more resistant than the other, initially the more resistant cell is going to be inhibited by the metabolites it's not getting from its partner, as opposed to by the antibiotic. And so, then what you'll see is that the more sensitive one will respond to the selection of the antibiotic and increase, while the more resistant one stays sort of static. At some point they'll switch, and then you'll see the other. I'm not explaining this very well, but one way to think about it is that in co-culture, if the two species depend on each other, you may be under selection to respond to the antibiotic less of the time, because if your partner is the more sensitive one, you're inhibited by the nutrients you're getting from that partner, as opposed to by the antibiotic. So as long as you had a scenario where two strains or populations were dependent on each other, I would expect those dynamics to come out. Hope that answers it to some extent. The feasible physiology simulations are great. Could you do it for any organism that has complete flux balance analysis done? Yes, so I should have explained that better. Could you do it for any organism? I'm going in the direction of the minimal cell Zanluthi-Schulten, published it for SIN 3.0. Would it be possible to use comments for either predicting SIN 3.0 morphology, or even competition between SIN and natural cells? Comets, you can, as long as you have a genome scale metabolic model, it can be incorporated into comets. You can run simulations with up to hundreds of species and get these quantitative predictions out. If the Luthi-Schulten paper I'm thinking of, I guess there are a couple there. So one, they did a cellular automata. So one thing to note about ours is it's not a cellular automata. The bacteria in a colony, we're actually tracking them as a field as opposed to individual cells. So we get a total amount of biomass, and then we can convert that back to thinking about individual cells, but it's fundamentally not a cellular automata. And that saves us some computational time, which allows us to do things like incorporate more species. So I should know more about the Luthi-Schulten models. I think some of them are more, more complex, but to the extent that they have just a genome scale metabolic network, then yes, we could easily incorporate that into comets and compete, a simple metabolic network against networks from whatever other species you wanted. What is the limit for Comet software? How many species can you analyze at a time? On my laptop, I've done up to 100 different species. Your computational time increases less than linearly as you increase the species number. Actually, what ends up slowing it down more is space. Diffusion calculations are a little slow. So if you get really big spatial landscapes, then it can become harder. But yeah, it is feasible to do hundreds of species, and it does them relatively quickly. So in in liquid. So if you assume that everything is well mixed and there's no diffusion, and you want to calculate, I don't know, 48 hours of growth of a complex community, that can be done in minutes. So the calculations are quite quick. We've only started playing around with that. Obviously, there are lots of constraints to comets, and the predictions it makes are going to be worse if the models you're putting in are not as good. So we've sort of focused on these ones where we can really rigorously test what's going on, and haven't done as much in these simulations with hundreds of species because it's harder for us to directly test the predictions that come out of them. But it absolutely can be done and make these testable predictions. Could you influence the colony size co-culture experiments by having one nutrient that is the limiting one and change the diffusion rates of that nutrient, for example, by changing viscosity of the media? This would be a cool experiment for a general physicochemical investigation of growth rate limiting factors. Yes, you certainly could. So again, that result is largely driven by the ratio of the growth rate to the diffusion of the resources. So if you change the diffusion of the resources, that changes the ratio and you'd see an effect. Functionally, we've found that harder to do. So for example, doing things like increasing the agar concentration in plates doesn't do much to change the diffusion constant of metabolites. So we've not done as much testing in that direction. But to the extent that you could change the diffusion of metabolites, then I think that it should actually absolutely work, and should generate these dynamics. Could you give us the remaining challenges to predictively modeling community metabolism, including metabolite exchange and population dynamics in the real environment? Yeah, so there are many challenges to just sort of flux balance analysis in general. One challenge is you need a good metabolic network to start with. And so for things like E. coli and Salmonella, those metabolic networks have been really well curated. For species that we know a lot less about, the metabolic network that we can create for those species is not going to be as complete. Others have developed really nice pipelines where you can start with a genome sequence, feed that genome sequence into the pipeline, and it will create the model. But the question is, how accurate will that metabolic network that's created be? And because it's metabolic networks that are inputs into comets, if the network you're putting in is not accurate, that will make the predictions less accurate. Additionally, flux balance analysis depends on this optimization. So you're doing a linear optimization, and typically what people ask the cells to optimize is biomass production. But we know that cells often aren't actually optimizing the amount of biomass production. And B, even in cases where they are optimizing their biomass production, if you put them in some environment they've never been in, it's sort of fundamentally an evolutionary optimality model. And if you put it in a novel environment, there's a reasonable chance that it won't be behaving there optimally yet, even though it might ultimately. And then some additional components are that flux balance analysis it's stoichiometric based. So it's a sort of glossing over many of the kinetics of reactions inside of a cell. So we do have Michaelis-Menten kinetics encoded on the outside, but on the inside, we don't have those kinetics. We can do things like put constraints on the flux through any given individual network, but we often don't know those. Additionally, the uptake, we can put in values for the Michaelis-Menten dynamics for every metabolite, but we often don't know that. And so we often have to make some approximations. So I actually started with a flux balance analysis thinking it was garbage. And so did a project in my postdoc to show that it didn't work for all these reasons. And was a bit surprised how well it often did work, that a lot of these caveats can be, that they are small enough that they often don't perturb your prediction that well, or that much. But fundamentally, I think of FBA as being most useful as a null model, where it says, if we understand the metabolism, and we can accurately define the nutrients in our system, and metabolism is what's driving the interactions, then these are the species ratios you should see. These are the rates at which carbon should be consumed. And to the extent that your experiments deviate from that, you know that there's some cool biology, and you have sort of a list of assumptions to directly test. So those are at least some of the constraints on it. An additional constraint in the current version is that there's some things like toxicity or antibiotic production which aren't incorporated, but we actually now do have that encoded and hope to be getting that out soon as well. So I hope that, yeah, I hope that provides some sense of some of the challenges.
Yeah. So that's wonderful. So, but my additional question is, I mean, beyond the individual cell, if we kind of want to understand entire, consortium of the bacteria or microbe to understand the metabolism and then including exchange of the metabolism, you are saying because right now individual metabolism in the one cell is not perfect, even considering entire community is very, very challenging.
Yeah, so I mean, I guess the answer is it depends on the community that you're interested in. So we were able to show that Comets was able to make accurate predictions about our 3 species community. And so with sort of simple communities, I think it will make more accurate predictions. As the communities get more complex, you're right that there's sort of compounding effects of errors. So if you have one species that is behaving incorrectly, then that will absolutely throw off your quantitative predictions. So with complex communities, I would not expect the same degree of accuracy in the predictions that are made. But I think that it could still make really useful qualitative predictions at the very least, in terms of sort of the relative proportions of communities or of populations within a community, how communities will be impacted by changing nutrients. So I think those predictions could be made. How accurate they are, I think, is a testable question that we haven't tested yet.
Okay, wonderful. Thank you.
Yeah, thank you. And I guess I should say that we are starting to try and extend this in other directions and test it with collaborators that work in more complex communities. But we're still sort of in the early stages of that. Could you apply predictive algorithms to design a new organism? For example, to engineer a new extremophile? So yes, one could certainly use flux balance analysis to, and this has been done a fair bit, to think about what sort of network you would need in order to consume a certain resource or secrete a compound of interest or carry out a given function. And one could then also extend that to think about not only how would you design a single cell, but if you wanted a consortia to carry something out, there are programs that have been generated by other folks, such as OpsCom, out of the famous lab that I'm going to blank on embarrassingly at the moment. But other tools have been developed to extend this to think about how you might design consortia for specific interests. I guess for the extremophile, some of the challenges could be that environmental perturbations, so things like temperature, can be difficult to think about how to incorporate into this model. So you can do things like change the Vmax of uptake, but in terms of changing like the stability of enzymes, for example, the comments wouldn't, or flux balance analysis wouldn't necessarily predict that. So yeah, so I think you could use it to make some predictions about designing novel networks. But yeah, I think it would depend a little bit on what aspect of novelty you were most interested in. Also, given the heterogeneity of the soil environment, the metabolic prediction of community dynamics and metabolism would be more challenging. Could you comment on this point as well? Yeah, and I should have included that slide. That's actually a direction that we're really excited about going. So we have a collaborator here, Linda Kinkle, and Mike Smansky, who work a lot on streptomyces in soils. And so we're excited about trying to model those. And actually, one of the cool things in comments is that you can define the environment in which all these simulations occur. So you can create very complex environments where, for example, you have grains of soil through which diffusion doesn't happen. So you can create these complex diffusion matrices or sort of complex series of channels through which diffusion can occur, and then set up simulations in this sort of synthetic soil where there is this complex array of space through which metabolites are diffusing. You can set up an oxygen gradient at the top so that the oxygen diffuses down through the soil. And you can even do things like there are now some, well, you can create plant roots. You can say that there's a root which is providing carbon at some place in the soil, and it is then diffusing out through the soil as it's generated by the root. So I think that we're really excited about trying to get at some of these more complex environments and using comets and other tools to try and make some predictions there. So it for sure will be complicated. And one of the big challenges is that we don't really know what that structure necessarily looks like in a lot of soils. But I think we can start making some predictions, at least, about the behavior that should emerge under different sorts of environmental heterogeneity. So I think, yeah, I think there's some fun potential there.
Yeah, that's cool. I mean, actually, I got to know about those tools from Dan at Boston University, and we tried to do some collaboration. But after I just watching your talk, it was fantastic. So that's why I need to ask that question, because I wanted to get some insight from you. So this is amazing.
Oh, thanks so much. Yeah. Well, that's great that you got a chance to interact with them. They're really fun.
Yeah.
And doing
Thank
cool
you.
stuff. So, yep, thanks.
I want to probably reach out to you later to discuss more.
Yeah, that'd be great. That'd be great.
Yeah, thank you.
So the paper, maybe I'll come back to in just a second. So there's a question on adaptation in co-cultures shouldn't be faster, stronger because of, for example, stronger effects of antibiotics. As I understood, bacteria in co-culture are more exposed to the stressors. I see. So what we showed was that that bacteria were inhibited by lower concentrations of antibiotic when they were growing in obligate cross-feeding co-cultures relative to when the bacteria were growing alone. And the reason for that is that because if you're part of a cross-feeding network where each species relies on each other, then any concentration of antibiotic, which takes out any member of that network, will cause the whole network to collapse. And so that was the reason that we were saying that that we showed that antibiotics can have stronger effects in co-cultures of cross-feeding bacteria than when bacteria are growing alone. Yeah, I hope that answers the question. Yeah, if not, definitely happy to take another stab at it. Oh, great. Okay. Great. And then let's see if I can. The paper for essential metabolism for a minimal cell was put in the chat, and I'm just taking a look at that really quickly.
I can shoot you an email with that link too. It's just a really cool paper, and I think that's the one that Chris had in mind when he asked.
Okay, and so they do create a genome scale metabolic network of the cell, but then are they doing? Oh, I guess I don't understand.
My understanding is it was explained to me that it's basically a flux balance analysis for this in 3.0, but I'm not an expert, so I don't know what flavor they meant. But yeah, it's just a really cool paper. Cool.
Yeah, I'll have to dig into that. I've definitely have seen a bunch of their work before, and yeah, they do just beautiful work. So, and they developed a program which was also very similar to Comet's, but used individual cells, which is exciting. So yeah, it'll be fun to see what they did here.
All right. I think that's all the questions.
Great. But yeah, thanks so much for giving me a chance to share some of our stuff with you all. And yeah, and it'd be great to follow up with anybody that's interested some more.
Thank you so much, and thanks everyone. And have a great rest of your day, and see you next week. Bye.
Thank you.